Today, in my inaugural lecture, I will describe how we can use gene expression as a guide towards disease prediction and personalized therapy. And I will use lupus as the disease example. So today I will present two projects that represent some of my research work over the last years. I will present a novel pathway that mediates kidney injury in lupus and a transcriptomics analysis, a transcriptomic analysis performed across tissues, across species, and across time. So let's start with what lupus is. Systemic lupus erythematosus is the prototypic systemic autoimmune disease that is characterized by loss of tolerance to self-nuclear antigens, leading to autoantibody production and immune complex deposition and damage to multiple organs, including the skin, the joints, the kidneys, the lungs, the heart, and the nervous system. It mainly affects young individuals of childbearing age, and is a highly heterogeneous disease in symptoms, severity, and response to treatment. The pathogenesis of the disease is very complex, and it involves all these type of cells. It involves cells of the innate immune system, of the adaptive immune system, and cells of end organ tissues that are damaged by lupus, for example, kidney cells or brain cells. Despite many scientific efforts, the pathogenesis remains elusive. Treatment remains empirical, composed of non-specific immunosuppressants, and most important of all, morbidity and mortality remain increased in this young adult population of patients. So why should we study gene expression that is synonymous with RNA in such a complex disease? This is a cell, and we all know that genetic information is transferred from the DNA to the proteins through the mRNA. And traditionally, a gene was defined as the region of DNA whose mRNA encoded the protein. All the rest DNA was not thought to be transcribed into RNA and was named as junk DNA. However, we got the results of the ENCODE project, whose aim was to identify, describe, and catalog all genomes, junk DNAs and gene DNAs. And the results of this project, not only did they demonstrate that this junk DNA is transcribed into RNA, but the results also demonstrated that the majority of variables, the majority of polymorphisms that are associated with complex diseases are located in these non-coding regions. Therefore, we understood that coding genomes, as well as non-coding but regulatory genomes, are important in complex human diseases such as lupus. And if we want to understand complex human diseases, we have to study and understand all genomes, junk DNAs and gene DNAs. And how can we do this? Through genome-wide high-throughput studies. In other words, through omics. Omics are non-hypothesis-driven and unbiased approaches that measure multiple entities simultaneously. For example, proteomics measure proteins, metabolomics measure metabolites, and transcriptomics measure transcripts. Transcriptomics measure RNAs. And then computational analysis compare differences of entities between conditions. For example, computational analysis compare differences of transcripts of RNAs between health and disease, or between cells, or between tissues. And this is a good way, at least in my opinion, to identify particular molecules, particular genes, particular proteins to be targeted in particular patient subgroups. So, to understand the pathogenesis of lupus, we elected to perform gene expression studies with transcriptomics. And we performed transcriptomics with the traditional method of microarrays, a hybridization-based technique. And we also performed transcriptomics with RNA sequencing, next generation sequencing technology. And we initially studied gene expression in the blood and the bone marrow from patients with active and inactive lupus. And we observed that molecular defects were tracked back to the hematopoietic stem cell, to the hematopoietic progenitor cell that lives in the bone marrow. And interestingly, we also observed that patients with active lupus and not in active lupus, were characterized by a strong neutrophil and an abnormal autophagy gene signature. And what is autophagy? Autophagy is a Greek word meaning eating myself. And let's suppose that this is a cell. Under conditions of cell stress, for example, under starvation, under hunger, cells break down their own components. For example, they break down their mitochondria to gain their energy and survive. And autophagy is involved in several cellular processes, including the immune response. And let's go to neutrophils. We all know that neutrophils are a type of white blood cell that represent the first line of defense of an organism. And this cell is a neutrophil with its polymorphic nucleus and a cytoplasm filled with granules that contain enzymes. And this here is a bacterium. So once bacteria invade an organism, then the neutrophils are trying to kill them by eating them. Neutrophils phagocytose bacteria and they kill them inside their cytoplasm by the action of the enzymes. However, if neutrophils fail to kill bacteria, they decide to commit suicide. They decide to kill themselves. They decide to kill themselves through a novel form of cell death named netosis that is synonymous with net release. And net got its name from the initial letters of the words neutrophil, extracellular trap. And these nets are extracellular chromatin filaments, extracellular DNA filaments that are decorated with proteins and enzymes released from granules and cytoplasm upon a neutrophil's death through netosis. And on these net, bacteria are trapped and killed. So, to examine whether there is a link between autophagy and neutrophils, we isolated healthy neutrophils and neutrophils from patients with active lupus and inactive lupus. And we stained with red a marker of autophagy. And as we can clearly see, autophagy is increased, is upregulated in neutrophils isolated from patients with active lupus. And also these neutrophils seem to extrude in the extracellular compartment this material that upon staining with green and red for two markers of netosis, this material became yellow due to the colocalization of this color. Therefore, this is a net. So neutrophils from active lupus patients release nets. And when autophagy was blocked in our cell culture, these nets disappeared. Therefore, autophagy is the process that mediates net release in active lupus patients. And why is this important? It is because these nets are also decorated by bioactive proteins. For example, they are decorated with tissue factor, a protein activating the coagulation cascade, and they are also decorated with this pro-inflammatory cytokine IL-17A, interleukin 17A. And together, these two proteins on the net increase the migration capacity of skin fibroblasts. This is a primary skin fibroblast system. They increase collagen production, and they also increase thrombin generation in the absence of platelets. Therefore, these nets released by lupus neutrophils that are decorated by bioactive proteins mediate fibrosis and thrombosis in lupus patients. And interestingly, we also found these nets in kidney biopsy samples from patients with active lupus nephritis, active kidney damage, and also in the skin of patients with active discoid lupus, active skin lupus disease. And using inhibition and stimulation studies, we further demonstrated that this pathway is mediated by endothelin-1, a vasoconstrictive agent, and HIF-1 alpha, a hypoxia-inducible factor, that these two upregulate RED1 that mediates autophagy and subsequent net release decorated with tissue factor and IL-17A, leading to thrombus formation and fibroblasts. So for the first time, we described a novel multistep pathway mediating end organ injury in lupus, kidney injury, and skin injury. And the interesting thing is that this pathway can be targeted using multiple agents through drug repositioning. Bosentan inhibits endothelin-1, L-ascorbic acid inhibits HIF-1 alpha, hydroxychloroquine, plaquenil, you know it, inhibit autophagy subsequent net release, whereas thrombin inhibitors and secukinumab block the bioactive proteins on the nets to reduce end organ injury. So this was a summary of the first project. Let's go to the transcriptomics project. Lupus nephritis, lupus kidney damage affects about 80% of individuals with lupus. Therapy fails to induce remission in more than 50% of them, with up to 20% of lupus nephritis patients eventually developing end-stage kidney disease, meaning that they require hemodialysis for the rest of their lives to survive. Presymptomatic or silent lupus nephritis is an early stage in the natural history of the disease. It comes before lupus diagnosis. Interventions in lupus mice demonstrated that kidney inflammation and damage can be reversed. So we thought that preemptive therapy before the establishment of full-blown disease, may be a good therapeutic option in lupus nephritis. However, a kidney biopsy that is an invasive procedure associated with increased risk of serious adverse events, including hemorrhage and death, is not performed in presymptomatic disease. So, can the onset of lupus nephritis be predicted so that it can be targeted promptly? I will say yes, through machine learning. Machine learning is a form of artificial intelligence that, in simple words, can identify meaningful patterns in big data, including gene expression data, to be used to predict an outcome. And there are several categories of machine learning. For example, in supervised learning, a labeled data set of features, for example, a data set of brain images with and without brain cancer, are used to train our model, are used to train our machine to recognize features, to recognize cancer. And once the model is trained, unseen data are applied to it. For example, the model is applied to unseen brain images with and without cancer. And now the model knows how to make diagnosis, how to predict our outcome. The machine can tell us which patient has or does not have a brain cancer. Lupus affects all tissues in humans. However, performing serial biopsies, sequential biopsies from multiple tissues in humans is not feasible. So to identify the gene abnormalities in lupus nephritis patients that are exclusively observed within kidneys and not in other tissues, we studied the classical model of human lupus, the F1 lupus model. And we studied this, we sacrificed the mice at the nephritic stage, the symptomatic stage of the disease, and the pre-symptomatic stage of the disease. And from all mice, we collected brains, spleens, and kidneys. We extracted RNA and we performed RNA sequencing. And then we combined our two lupus cohorts. Thus, we created a large lupus cohort composed of 106 healthy individuals and 260 lupus patients. And some of these lupus patients had active lupus nephritis, some of them had inactive lupus nephritis, some of them did not have kidney damage, some of them only had joint or skin disease, some of them had neuropsychiatric lupus. And from all humans, lupus patients, and healthy individuals, we collected their blood, we isolated, we extracted RNA, and we performed RNA sequencing. And this is the largest to date human mRNA sequencing cohort. So to understand the mechanisms that are involved in the progression from the preclinical stage to the clinical stage of lupus nephritis, we compared gene expression profiles across tissues from lupus mice probed at the symptomatic versus the pre-symptomatic stage of the disease. and about 500 genes were differentially expressed only within kidneys and not in other tissue studies. And these genes were involved in the transition towards the nephritic stage of lupus nephritis. Previous data from other labs demonstrated that gene abnormalities are similar within kidneys from lupus mice and patients with lupus nephritis within their kidneys. And previous studies also demonstrated that gene abnormalities, gene alterations within kidney and non-kidney tissues, such as the skin, are also similar in lupus nephritis patients. So we questioned whether the mouse kidney-specific gene signature also exists in humans with lupus nephritis, but at this time in their blood. That is a tissue easy to collect and minimally invasive. And specifically, we asked whether the mouse kidney can serve as a non-invasive marker, as a not requiring biopsy marker of kidney disease in human lupus. So to answer this question, we first performed RNA sequencing in the whole blood of our human cohort and identified thousands of differentially expressed genes between patients with lupus nephritis and healthy individuals, and thousands of differentially expressed genes between patients with lupus nephritis and patients with lupus without kidney damage. And to examine whether the human blood and the mouse kidney share common gene expression alterations, we compared the kidney-specific signature of lupus versus healthy mice with the blood signature of patients with lupus nephritis versus healthy individuals. And we found that the majority of mouse genes also exist in the blood of this group of patients in this comparison. A similar cross-species analysis was performed to examine whether the kidney-specific signature from lupus mice probed at the symptomatic versus the pre-symptomatic stage of the disease also exists in the blood of lupus nephritis patients versus non-lupus nephritis lupus patients. And 97 genes were common between the two groups, and these 97 genes were involved in the transition towards the clinical stage of the disease across species. Early identification and prompt therapy in lupus nephritis are linked to improved outcomes. However, demographic, clinical, and serological data cannot predict the onset of kidney disease in lupus. So, can the mouse kidney genes and the human blood genes predict those lupus patients who will develop lupus nephritis in the future? To answer this question, we first splitted our combined mRNA sequencing dataset cohort into training set and validation set. The training set was used to train our machine learning model, and the validation set was used to validate our algorithm. And to examine whether we can predict lupus nephritis patients from lupus patients without kidney damage, we have used the kidney-specific signature from lupus mice at the nephritic versus the pre-nephritic stage of the disease, 500 differentially expressed genes. We removed noise, and using machine learning techniques, we selected the top genes based on accuracy. We then we constructed 6 different machine learning models, and models were fit to identify which performs best with the top selected genes. And the best model was selected at the end based on accuracy, sensitivity, specificity, and area under the ROC curve. And we were very happy to see that 20 genes, including age, gender, and the presence of anti-dsDNA, could predict our outcome, could predict the lupus nephritis patients from lupus patients without kidney disease, non-invasively, not requiring a kidney biopsy. This model could predict our outcome with an accuracy of 82%, low sensitivity, 63%, but high specificity, 90.2%, indicating that this is a good model to identify true negative cases. But I have to tell you that the same machine learning model could predict non-invasively and not requiring clinical parameters patients with lupus nephritis from healthy individuals with an accuracy of 96%, sensitivity 100%, 93% specificity, and area under the curve of almost 1. So, to sum up, from gene expression, from mouse gene expression, we were finally able, through machine learning techniques and genome-wide throughput studies, to predict human clinical characteristics, different human clinical characteristics, in a complex human cohort such as lupus cohort. Let's go to computational systems biology that combines knowledge-driven experimental data with simulation-based analysis to test hypotheses with in silico experiments. The Connectivity Map project was the first project to report genome-wide gene expression responses from 4 human cell lines, treated each time with one out of more than 1,000 FDA-approved drugs or small molecules at different doses. The links further expanded the CMAP project and provided more than 1.3 million gene expression profiles from more than 60 human cell lines that were treated with more than 20,000 perturbagens, and these gene expression profiles were given before and after treatment with these perturbagens. And this search engine, the L1000 CDS squared search engine that is free, online, further prioritized thousands of small molecules and their pairwise combinations according to whether they can mimic or reverse the gene signature input. Here we input our gene signature of interest. So, using genome-wide high-throughput studies, using computational analysis and computational systems biology, we can find novel drugs to treat the disease, or we can find drugs to be repurposed. Let's suppose that we have a disease signature here demonstrated with a green color. And let's suppose that we have a drug signature with green color and red color. Matching between the two signatures in the opposite direction can reveal a compound that may reverse the disease's phenotype. So to identify novel drugs or small molecule compounds that are predicted to reverse our gene signature of interest, we have uploaded to this search engine the 97 differentially expressed genes that were common across species and were involved in the pathogenesis from the preclinical to the clinical stage of lupus nephritis. And the top 50 drugs and their combinations were found. And specifically, these 3 small molecule compounds, an adenosine receptor agonist, an NMDA receptor modulator, and this histone deacetylase inhibitor, were predicted to reverse our gene signature of interest in this human kidney cell line. So to complete the puzzle, from mouse gene expression, we were able to predict human distinct clinical manifestations in our complex human disease cohort, and with the help of computational systems analysis, we were able to identify the right therapy to be given to the right patient at the right time. Of course, all these need to be validated and need to be validated in other databases and other mRNA sequencing dataset cohorts. So, of course, these findings and applications can extend beyond lupus and the kidneys. Since my ranking on June, 3 papers have been submitted and are currently under revision, and I hope they will soon be published. And regarding ongoing projects, currently we are studying the role of NETs in cardiovascular disease, and specifically in myocardial fibrosis. And these are hearts from lupus mice and healthy mice. We can see that lupus mice have larger hearts from healthy mice. And these sections have been stained by Stavros Doumas. He's an MD now at Georgetown University Hospital. And I have to tell you that this is a project funded by the Onassis Cardiothoracic Center. President of Onassis, Professor Boletis, is here with us today, and I thank him for that. And I have to tell the medical school that some of our students have already expressed their interest in collaborating on this project. In collaboration with Konstantinos Gambas, a researcher at the Hellenic Pasteur Institute, and in collaboration with Nikos Michail, a surgeon of the Organ Transplantation Center, we are now studying the role of nets in transplantation, and our preliminary data indicate, I don't know whether you can see it, we can find some nets in kidney biopsy samples from patients with T-cell mediated kidney rejection. I'm really looking forward to the final result. Some other students also expressed their interest in collaborating in this project. So today I've shared with you the kidney-specific story. Now we are working on the common cross-tissue and across all stages of lupus gene signature. And we are also trying to link the spleen genes with hemolytic anemia, autoimmune hemolytic anemia, and we are also trying to link brain genes with neuropsychiatric lupus. And of course, we are also interested in gene expression differences between females and males, not only in lupus patients, but also in healthy individuals. I'm sure that we will find many differences. And our transcriptomic analysis was mainly performed in collaboration with Panagiotis Garantsiotis. He's an MD now at Hanover Medical University, and I can assure you that in his title, next to the MD, we have to place an MVP. In my opinion, he's the most valuable player in our team. So, I would like to acknowledge all my collaborators, apart from those already mentioned, Nikos Michail, Konstantinos Gambas, Panagiotis Garantsiotis, Stavros Doumas. I would also like to acknowledge all my collaborators at Limassol General Hospital, Panagiotis Pedousis and Michalis Frangos, relatives, but artificial intelligent guys who helped me a lot through all these process. I would like to thank Konstantinos Rittis and Akrivi Chrysanthopoulos from Alexandroupolis University Hospital, Smaragdi Marinaki from Laiko University Hospital, and all my collaborators at the Biomedical Research Foundation of the Academy of Athens. I cannot name separately each of them, mainly Panagiotis Verginis, George Bertsias, and Emanuel Dermitzakis. These are our funds. Without their funding, nothing was going to be able to be performed and published. And of course, we all thank all patients and their referring physicians for collaborating with us in our research. And please allow me separately to thank my teachers. Aristidis Charonis, he was my first teacher in lab. He was the teacher who first believed in me. I would like to thank Ioannis Boletis, professor of nephrology at the Athens Medical University. Apart from nephrology and transplantation and glomerular diseases, he is the teacher who supported me and supports me endlessly. And last but definitely not least, I would like to thank my professor, Dimitrios Boumbas. He is a professor of medicine and rheumatology at the Athens Medical School, and he's the teacher who made me a scientist. He made me think like a scientist, write like a scientist, behave like a scientist, I hope. He's the one who gave me all opportunities, and I owe him every scientific achievement. And of course, I would like to thank the medical school for giving me the opportunity, and I wish we can collaborate, and I promise I will do my best.